Okay, everybody has a good lunch, I guess. For me, it is. I'm full. I don't know if I'll sleep also. It, it is a very good lunch. I really enjoyed this. Thanks to Dr. Reddy and Dr. Ashok.
We didn't do anything, yeah.
You took me there, right?
But yeah, we helped to get that.
Yeah. Okay, so I think we'll talk today's sessions on the pox virus, which is under myxoviridae. There's two myxoviridae, one is para and ortho, so this is the orthomyxoviridae. Paramyx is other viruses like rinderpest, Pistidipetis ruminants, those viruses are there. Canine distemper, those are all paramyx. Orthomyxo is mostly famous for influenza, and we all know that we have flu and all those. So there are a lot of viruses, but we are only focusing some of the important which affects the humans and also some of the animals, because as we all know that okay, thanks. As we all know that also there is a recombination happens. Probably you guys have heard the avian flu, swine flu, those things. So these came from swine. They came from, you know, chickens, poultries, and all those. So those, that's why they are very important. So influenza A, B, and C and D, they are very important. But A, B, C are major for humans. D actually mostly in the pig and cattle, and there's, I don't think, any other important. The thogotovirus is not too much taken care of such importance, but they are major is A, B, and C, and the more important is influenza A among those all A, B, C, and D, and you can see there are a lot of serotypes. You can see H and N. H represents hemagglutinin, N for neuraminidase. We'll come back in a second. What are those? And you can see the columns showing hosts. So these are the major hosts where it infects. So also, have you seen those? Orthos means for straight, and mixi means slime or mucus. So the mix, what it means that this virus produces mucus. Mucus means you have a nasal secretions and all those. So the virus causes affect the respiratory system. So that's why uh, mucus. So that's why the name is orthomyxo. This is another phylogenetic tree. Just want to show that the influenza A. There are several strains. I'll define how those nomenclature came from. In another slides. But just see, like they normally present A slash equine slash prig slash some numbers, then some year. So it is kind of influenza A, which species, where it has been isolated, and what is the types and the year when it is isolated. So that way they have defined all this influenza virus, because there are so many subtypes, serotypes. And A, B, and C, they are very closely related based on phylogenetic tree rather than the other viruses there. So here's the structure of the influenza virus. So as we said, H and N, which you see H1, H2, H1, N2, H5, N9, those things. So H represents hemagglutinin. So that is the surface proteins. And then the neuraminidase is present in between hemagglutinin. So that's the structure of the virus. They have hemagglutinin and neuraminidase all the time. And then they have a matrix protein. And you can see there are some uh, segmented RNA. So you can count those, how many segments are there, like 1, 2, 3, 4, so 8. So there are 8 segmented RNA there, and every RNA they have looks ribonucleic acids, which are RNA dependent RNA polymers. These are the polymerase complex, and also the nucleocapsid proteins, nucleoproteins presence there. And it is an enveloped virus. You can see lipid. So what does that means? Like the cover is coated with lipids. So that's why they say um, if you see, go to the market and they say, oh, take this spray, or it will kill all the flu viruses. What that means, because that chemical has something which dissolves lipid. So if it dissolves lipid, so the virus get rid of this lipid envelope because it's lipid soluble. So that's why they sell, any lipid soluble agents, and in the form of spray, they say, oh, take this, it will kill all the flu viruses. That means that reagent actually acts on it and dissolve this lipid envelope. If the envelope is down, then the virus is no more live. These are the electron microscopy pictures, and here you can see the size of this virus, and helical nucleic acid is a lipid envelope, and HA actually that helps for virus attachment. The virus attached to the celiac acid receptor. So every cell has a celiac acid, it's a receptor. So the virus bind with this celiac acid receptor and go to the inside the cells. The neuraminidase is another thing, it destroys the celiac acid, so to release the viral progeny. So when the virus's multiplication happens, the nucleic acid cleaves those celiac acids so that the virus can come out. So I said there's 8 segments. You can see 1, 2, 3, 4, 8, and they are different sizes, and they are linear, negative single stranded RNA, and you can see all the functions. PB means PB1. Those are actually non-structural proteins. They make, they are doing RNA polymerase. These are another RNA polymerase for cap recognitions. This is N40. This is also function unknown. But these are all non-structural. So structural comes when hemagglutinin. This is a structural proteins, nucleoproteins, neuraminidase. This is structural. Matrix protein. This is also structural. This is non-structural. So, and you see their functions and what they do and all those. But basically, just remember this influenza is a segmented virus. There are 8 segments are there. This is the replications. Again, same like if you remember the earlier slides, earlier lectures today and yesterday, the virus adhere to the cells, and definitely it needs celiac acid receptors, and then virus get in, and then virus after getting by encapsulated, the RNA is released and go to the nucleus, and then RNA makes so many copies, not only for their own, they also make mRNA where that all the structural proteins formation happens, or sometimes the structural proteins go in because there are also viral RNA there, so they the combinations of viral RNA and proteins combine together, and then they come out from the nucleus, and then again Golgi is another, I mentioned also, it is the cell organelles which actually with the packaging, everything packaging happening here, and then the virus released. Sometimes they also release. So yeah, what I'm saying, the hemagglutinin and neuraminidase, those are released because those are the structural protein they express, and the whole package comes in, and then the virus released there. So hemagglutinin, the more important for the influenza, flu vaccine, or anything about flu is those 2 important things, hemagglutinin and neuraminidase. So based on those, there are a lot of combination or serotypes happening. So H1 to H7, there are 17 different proteins, and N1 to N10. So why they are numbered like this is based on the structures as well as the how many amino acids they can produce on those things. So H1 and H3 considered the human strains, same like N1 and N2 considered human strains, but the rest mostly they found in cattle or pig and chicken and all those things. We mentioned already that hemagglutinin binds to the host cell receptors, which is actually celiac acid, and this is actually clips polysaccharide chains from the host cell surface, actually that breaks the celiac acid and facilitates new virus. So everybody have a flu at some point, right? Or they don't know, or somebody knows. Because flu always happens 1 to 3 days incubation period. And these are the symptoms you should feel: high fever, prostration, headache, myalgia, body ache, pharyngitis. You cannot talk too much. Your pharynx, you think something stuck in your, and you're coughing all the time. So these are the common symptoms when you have flu, and this is the flu season now, and it is very common. The complications, flu actually also calls the pneumonia because it's influenza. Uh, so it's more risk is young children, elderly peoples with chronic pulmonary disease. Maybe those are taking a lot of smoking, some tuberculosis patients, some chronic disease they have in the pulmonary lungs. Young children also suffers the same way. Causes is direct effect of influenza virus. Immunopathogenesis means, immunopathogenesis is a big term, means how immune cells interacts with the virus and causes the pathogenesis. So it is a complicated cascades of mechanism when virus infects to the cells, and how the cells react by releasing different cytokines, chemokines, or other growth factors, even like different anti-inflammatory or pro-inflammatory cytokines. It may also accelerate the complications if we have a secondary bacterial infections along with that, or other superviral infections. So if I have a flu, I also got TB, then I'm very serious condition. I may have go to hospitalized for several days or so. Same like flu I had happen, and I'm traveling. I got COVID again. It's a very serious condition. So flu itself a bad disease, and then you get other infections that's accelerate the pathogenesis. So other complications also happens. Ray's syndromes, which is rare, but it is a serious illness of the liver. Somehow it affects the liver of those patients who got the flu. The exact cause is unknown. They always say you should not take aspirin, because aspirin, do you know aspirin does? Aspirin actually more used as a pain. It's a painkiller. It's a very old medicines came, but it's also good for regulating your blood pressure, and all those people use for aspirin. People have a heart attack or something, immediately take aspirin. It's like lower dose. It's 81 milligrams tablets you can get easily in the market because it's very cheap. It's very good drugs so far discovered by the human beings. Aspirin is the best drug. But there are a lot of drugs came out for the heart disease patients. But aspirin has been being used. A lot of heart patients used aspirin regularly because it also prevents clotting. So, but for flu, they are not recommending take aspirins. Maybe it's accelerate the hemorrhages. That's why. Um, Guillain-Barré syndromes, that is another autoimmune disease. This is we can discuss later on what is Guillain-Barré syndromes. It is an autoimmune disease, generates probably autoimmune always happens when your body generating antibodies unnecessarily, like especially IgD. There is another immunoglobulin which I never talked before. IgD and IgE, they are major source, especially IgE is the major source of generating autoimmune disease. Somehow in flu infections, IgE production increases in some patients, and that may lead to Guillain-Barré syndromes. And then there are the other clinical symptoms you can see, like severe weakness, numbness or paralysis in legs and arms. But it is very rare. In 2 in 100,000 infections, it's very rare, and fatality also very low. So just for your information, there are complications there also in influenza. It's not like its symptoms it's go away, but some patients or some people may have to face those consequences. Diagnosis, it's we talked about how to do diagnosis. Always the gold standard is virus isolation. If you go, I know very well. I don't know how doctors do here. Like if I have a push, especially now, all the schools in the United States, I'm talking my daughters. She has a cold and a fever. We took to the doctor. They're like emergencies sections, like not hospital emergency, but they have a separate also. They have a emergency, which is open till like about 20 hours per day. So they close very late. They are open early. So if you have an emergency, you go there. They will take a nasal swab, like COVID, you probably know. Always so they take the nasal swab and then do the test. So it's kind of lateral flow experiments. You just put it there, and they have a machine also which they do CBC. So it's a complete blood count. So they take the complete blood count immediately from the blood. They took a blood, small blood, and so they just tell you how much your neutrophil level is, how much your other white blood cells is, how much your RBC level is, whether you are anemic or not, whether neutrophil influx is happening or not. So neutrophil is another thing. As I said, like neutrophil platelets comes first, right? They are the first safeguarding, like a border. If the Pakistan hit us, like they are border people, they fire first. Then the army goes from New Delhi or somewhere where they're stored. The reserve people, reserve army goes later on. So the neutrophil also in the border line, they go first. Platelets go first. So they immediately test those. So the rapid examination, so that's they do. Take the swab, sometimes take the throat swab. So that's why they find it out based on the antibody test. Oh, you have this. These are the, very, I think it's a very old test. We did long when I was doing my master's graduation in egg. We can inoculate in the egg and then change. You have to be like a, how is it? It's a live egg, okay? And it's you are hatching. There will be hatcher. You just it's an embryonated egg, and you just put it there and then see the change of the embryo inside with the like that kind of light, with the hazel light, and see there is any hemorrhage happening or not. You can Google it, can go to YouTube, you can find it. But this is a very old technology. It's still going on in the embryonated eggs, and you can grow a lot of virus from this. So these are the old technology, but virus isolation is always a gold standard to find out which virus it is. And especially the cell culture in eggs is very, very unique and very conformative. PCR, as we all know, is mostly important. Serology test, hemagglutinin inhibition. This is another important test. To find out whether the hemagglutinin, which is present in the virus, how much antibody they generate. So if you have an antibody already standard, you can test how much virus is there. So by doing the hemagglutinin, we can talk later. It's a very nice. You can type it in Google, hemagglutinin agglutination assay. It will tell you how the assay is. It's very fancy assay. It's very old assay. And the inhibition assay is another way to confirmatory diagnosis. And ELISA also looking at IgG, IgM, those things. Okay. Treatment is more effective in first 36 hours of disease. Like as soon as you saw the symptoms, it's better to treat them. There are some drugs already there. Like M2 inhibitor. M2 means it is affecting on the matrix protein of the virus, but it is active against type A only. But there are other amantadine, rimantadine, virus resistant. So this antiviral is already discovered long ago. When as a student I was reading, so it's very effective anti-influenza drugs still available and works very good. There are new form like Tamiflu, which came little later. These are the older neuraminidase inhibitors. It is active against both type A and B, but here you have to type it in. But type A is more common. So just people, doctors before they diagnosed in depth, like whether it's type A or type B, they just prescribe those. But if you have both type A and type B, the Tamiflu, which is another oral drugs, which is very good. Or also people use the Janimivir, which is by inhalation. So you just take a nasal inhaler and just put it there, and then this is very also effective. So, when people sneeze, do you know how far it goes? So if it is a strong sneeze, it can go to 6 feet. So, they have studied a lot about the sneezing and how far the organisms can travel and still viable. So, from this area to this area, the organism right here, it goes about 6 feet. And so that's why, probably you have heard in the COVID, 6 feet distance everywhere they were marked and all those, because that's the reason, like, when you sneeze, the droplets can carrying the virus can goes at least 6 feet. That's the maximum. So flu epidemiology: flu spreads rapidly in seasonal epidemics. Okay, it is every year you'll find out there are flu somewhere. 5 to 15% of the world each year has the flu. 3 to 5 million cases of severe disease, and every year 250 to 500,000 deaths annually. So think about how badly the disease is. Mortality higher in elderly, so that's why it's always recommended, especially in I don't know Indian government, but United States, it's always mandatory to elderly persons should be vaccinated. Children's may skip, but the school says, school also we have to report, like every year when they go to the next class, we have to submit the vaccination report. And if something wrong, they say, oh, you have not taken flu last year, why? And you have to give the explanation. So you have to say, oh, you're traveling, or you cannot catch up. But the flu is free to the government. US government has given flu to every, and it's free. Most of the vaccine are actually free for children's, for adults. Special vaccines, if you are traveling, like if I'm traveling to Africa and I need yellow fever, then if you say it is important, it's job related, and although then it will be covered by insurance. But mostly it's all vaccines are free. And flu is another one which is mandatory for kids going to school. Yeah, and that is should be taken. And elderly also, it's mandatory. Pandemics due to antigenic shift occur less frequently. So just focus on this. So this is our globe, right? And this is the northern hemisphere and the southern hemisphere. And this is the tropics, right? So the north, and we are India is in between the tropical area, right? So if you see the northern hemisphere, because the climate change, cold and hot, it's extreme. So the peak happens and then it goes. So the influenza activity peaks very early and like 3 7 weeks and then go down. But in India, it's year round. Whether it is January, of March, or April, or May, always flu exists because the temperature and all those, the virus is very prominent. So that is the case. And here also you can see the southern hemisphere is another way. Influenza peak is April to September, and our case is November to March. So in United States, which is right here, right here. So they always prefer, our vaccine starts November, and we always take November, and so that it can cover the peak time. And for the other part, like southern hemisphere, mostly Africa, they those are the peak time they get the vaccines. But for India, it's there's no time; it's always there. So there is no peak, always peak. So I told you like why the name nomenclature came like this. So A for genus, whether it's influenza A or B or C or D, and the Hong Kong is the geographic regions where they have find out. This is the strain number. The whether it is E something or some, so it is like 1073. That's the strain. It's the 99. It's the year isolated, and hemoglobin type and neuraminidase type. So based on that, it says HN or N2. So you can see, if it is an article or newspaper, when they mention like this, oh, you got H9N2, then you remember those are actually they're talking the virus specific to hemagglutinin, which is the 9, because it starts from 1 to 17, and neuraminidase 1 to 10. So it is the number 2 and number 9. And if they say the whole thing, you know, oh, this has been isolated on Hong Kong long ago in 1999, and that virus is still present. So when people do like a flu screening or phylogenetic tree, like, you know, maybe this year, you know, some person died. You know, I'll show you some slide in New Delhi. There is outbreak, so they can isolate the virus, and then they can match it off, whether it is the Hong Kong virus or, you know, Spanish virus, or it has happened in India before. They have isolated, so you can match it up that the virus is coming back or not. Make sense? So interpandemic influenza. So due to the mutations that occur during replications, antigenic shift. So I'll show you the video very soon, and so you can understand what is antigenic shift. So that is the important part of this influenza virus. Why there is a different kind of serotypes. How H1N1 suddenly becomes H9N2 or H5N2. So this virus is very smart. They can do those antigenic shift, and they can be more virulent. They know how to safeguard themselves. And this video will talk about antigenic shift, and because that is the variation in hemagglutinin and neuraminidase proteins. So these are, as this is changing, is the antigenic drift is happening. That is responsible for seasonal influenza, and new vaccine developed each season. So that's why, because it's not like, you know, 20 years ago or yes, last year, oh, we have H5N2, so we are fine, next year will be the same. No, because the antigenic shift is there, and the virus changes based on, the virus has to modulate themselves to survive in the host, and they change it. And can you please play the video? So this video will talk about how it happens.
Neuraminidase, it removes oligosaccharide component, glycolipase.
Mm-hmm. Yeah.
Generally, it's
Yeah.
there on the cell.
Right.
So it makes easy for the virus to make it.
To make entry, yeah.
Circulating flu strains may change and make the old vaccine ineffective. A new vaccine is needed. To match the new viruses. The heart of a flu virus has 8 string-like segments called RNA. These are the genes that encode parts of the virus. Around the RNA are different proteins. The proteins involved in drift are called hemagglutinin and neuraminidase. Scientists call them HA and NA. There are 16 subtypes of HA proteins and 9 of NA. From these protein combinations, scientists name influenza A viruses.
Video.
For example, an H5N1 virus contains hemagglutinin subtype 5 and neuraminidase subtype 1. If a person has been vaccinated or has a natural immunity against the virus, antibodies will attach to the HA protein, blocking it from infecting human cells and stopping the virus from spreading. Without natural immunity, or immunity provided by the correct vaccine, the HA proteins on the virus interact with sugars on the outside of those cells. This creates a bond that allows the virus to enter targeted cells where it can cause infection. When the influenza viruses replicate, they can mutate slightly. Scientists have shown that this leaves small changes in the HA and NA proteins. Over time, as the virus spreads from person to person, these changes accumulate and the virus drifts from its original form. Eventually, the immune system no longer recognizes the HA proteins from the virus, and the flu vaccine becomes ineffective. Each year, a flu vaccine is made to protect against the most common flu viruses predicted to circulate among the population. Historically, scientists have done a remarkable job of matching the vaccine to the circulating viruses. Although there is an effective vaccine, not enough people choose to get it. As a result, influenza sickens between 5 and 20% of the U.S. population each year. More than 200,000 people may be hospitalized, and up to 49,000 people may die. That is why health officials recommend flu vaccination before every flu season.
Okay, so you understand how the drift happening. Yeah, so.
So does the virus have all these genes that are meant to code to different HA and NA proteins, or does the mutation happen and it happens to become one of these other things?
So mutation happens inside. So like if the I'll talk later in a couple slides. So the virus is there, like I got the virus, same H1N2 something. Then the virus, I produce antibody against H1N2. So the next year, or maybe the same, like H1N2, they mutate. There are two kind of mutation happens. One is antigenic shift, antigenic drift. So antigenic shift is I'm changing from H1 to H3. That is a shift, and sorry, that's a drift. And antigenic shift means where there is a mutation within the neuraminidase. So instead of N1 or N2, I now have different mutations, which is N5.
New type.
Yeah, new type. So then the antibody is recognizing only that H and N, those antibody, but now it is totally new and those are useless. Those antibodies are useless. So that is happening for flu. That's why every year they it's mostly kind of a machine learning and looking for algorithms of what happened last year, how different strains are available, how many different people got infected. So they do all kind of genotyping, then they come out to, okay, they make a mixture. I'll show you that in a couple slides below. And then they make a cocktail vaccine. And so that it at least can protect some of them. So it's not 100% protections, but better, as the slide says, like if you don't take it, there are more chance you get more sick. And it happens to me, like one time I just escaped and just try, like, okay, I'll not take vaccine. What happens? I was really bed sick. I cannot get up bed for the 2 days. It's the whole body pain. I have a fever and everything. So my wife and kids all took except me, and like a guinea pig, right? I tried myself, and it was very sick. So since then, I always take, despite it protects or not protect, at least it gives partial protections at some point, because that is another immunology subject, right? We are also studying those, and several others have studied, like non-specific immunoglobulin response also some way protects. Like if you have there are some non-specific antibody, which is not specific to there are two kind of antigen specific and non-specific. The non-specific is very important. You probably heard Rafi Ahmed, who is a very good scientist. I think he is from what is that university? Yeah, in India. So he's a big immunologist. He is in the Emory University. I forgot the university name. He did his PhD or something. And he is a big immunologist, and he has shown several papers in another model which is called LCMB models, and they have shown the non-specific antibodies, another very important thing to protect initially. I think it is still happening for flu virus also. Like though you have a vaccine-mediated response from whatever vaccine you are taking, for example this season, though I am getting infection from another virus, but I have already generated some non-specific antibody, and that antibody acting as a non-specific antibody for the new virus, right? So that gives some protections. And Rafi Ahmed, which is a very big immunologist, is in Emory now. So he has shown fast that non-specific antibody is very important for protections in the initial stage before the adaptive immune response, which antigen specific comes. That gives protection more systematically. Next slide. Okay. So here is I'm going to say like how the things changing here. So there is a lot of reassortment when two different viruses infect one host, which is called genetic shift, okay, or you can antigenic shift, what I mentioned. So antigenic drift, and so like for example the duck, okay, there are like H5N1 and human H3N2. So the pig somehow, maybe this is a like an animal farmer, and he has H3N2, and he has a pig farm. The pig got those virus, and the duck, he has also duck in the backyard, and then the duck is also infected, and pig also got the duck infection. Then there is a reassortment happening. So you have all those D1, D2, all those here, and human H1, H2, all you got here, and then it has a different kind of version you got, and then there is a reassortment happening. So that it changes, it has a new mutations, because you see H4 is more prominent now, and you have a DCA, D7 is prominent, D2, so rest kind of is gone. Like H1 is no more here. Okay, so there is a reassortment happening. Make sense?
This happens inside the body.
Yeah, inside the body. So this is kind of because that pig got co-infection, okay, and then the virus mutates themselves because two different viruses they are sharing each other. Okay, I'll take your H, I'll take your N, and the rest gone away, and then they make a different kind of combination here. So it's called reassortment. Then it can easily infect the another persons, and then it's maybe the virulent one.
Is that like a segment of the RNA getting shared?
Yeah, because I mentioned like influenza has a 8 strand, so they can share. I'll take your N protein, okay, rest I'll keep it. Oh, I'll take your M and H, take it, keep it. So that way they change. It's a lot of combination, right?
Yes, all sorts of permutations and combinations.
All kind of combination.
But only thing is that which is kind of like virulent, that only.
Exactly.
So it has to infect one cell has to be infected by both.
Of course, yeah. That's why the reassortment happens. So the cell is infected by all three, and then it makes different, making a mixture, and which is more pathogenic, we only care about, right? The less we don't, like your question yesterday. So yeah, the less is there, but they are less pathogenic. They are, has nothing to do. But then you got, so this is very, very unique to the flu virus.
So sometimes it is a random packaging also takes place, right?
Yeah, it's a random. We cannot predict. That's why we cannot predict. Even though we have not a vaccine that can give lifelong protection, no way. Because every time they change, every time it's changing, and we cannot predict. We can, because it has a 8 strand, and you also don't know how many virus they got infected, right? We are giving just a simple example. It may got from duck or may human. Maybe you got from another poultry. Maybe you got from another pig. Who knows?
But I also wonder that if the genome is so small and the required number of proteins and whatever antigenic variants that are limited, so how many ever combinations are possible? That is like in the more virulent ones, combinations will be very, very minimal or less number.
Yeah, so that's why I'll come in slides another. Like you can see there is a trend that these are the more pathogenic, because it has 8 strand, okay, 8 different segmented, because 8 segmented RNA, and now it is how many multiplication combination, and also you have to consider H is 1 to 18, numerate is 1 to 10, so you have to also in your mathematical calculation those combinations. So 8 times 17 times 8, 10. So that many different combination permutation it may happen. It's a huge number though.
Permutations and combinations are few, definitely.
Yeah.
But each protein or anything to be virulent or something, it can go only undergo limited number of mutations because the protein cannot undergo so many changes to be virulent or something.
No, but
kind of like non.
That's true. But there is also reassortment happening, right? I take the H9 or H5, which is virulent. We know H5 is virulent, and then I got N1 from the duck. So it may be very.
Both the proteins they are going to be more viral, like if they are going to be there in one time, so probably be more effective.
Yeah, exactly. Yeah. So then results is, so there is another video, results seems completely different virus unrecognized by the, so because there are a lot of reassortment happening, so the human system, the immune system cannot recognize. So you can play the video here. Um, yes.
Pandemics, or global epidemics of influenza, have occurred for hundreds of years. There were 3 pandemics during the 20th century. They occurred in 1918, 1957, and 1968. The 1918 pandemic took the largest toll, causing an estimated 50 million deaths worldwide. The first pandemic of the 21st century began in 2009. But how does a pandemic occur? Influenza A viruses vary slightly from year to year, a phenomenon called antigenic drift. These small variations are the reason why the vaccine must be updated every flu season. Influenza A viruses also can undergo dramatic genetic changes called antigenic shift that may cause a new influenza strain to which we have little to no immunity. If this strain spreads easily among people, it may cause a pandemic. The immune system targets 2 proteins on the surface of influenza A viruses, hemagglutinin and neuraminidase. Hemagglutinin has 16 confirmed subtypes of proteins. Neuraminidase has 9. From these protein combinations, scientists name influenza viruses. For example, an H5N1 virus contains hemagglutinin subtype 5 and neuraminidase subtype 1. Antigenic shift can occur when 2 different influenza A viruses circulating in the environment infect and multiply inside the same cell. While inside the cell, these parent viruses can exchange or reassort genetic material and create a new virus. Through reassortment of the 8 gene segments of each virus, there are theoretically 256 possible virus combinations. Pandemic viruses also can emerge in other ways. Among these is adaptation. Scientists believe that the 2009 H1N1 virus was the product of 2 influenza A viruses that were circulating in pigs. The viruses are thought to have reassorted and adapted to create a new virus capable of infecting people. If an emerging virus transmits efficiently from person to person, it would be a challenge for scientists to produce an effective vaccine fast enough to limit its spread. That is particularly true with the amount of international travel that occurs today. In a matter of hours, one person infected with a new form of influenza can spread the virus to people in another part of the world. The National Institute of Allergy and Infectious Diseases and other research institutions around the globe are working to find better ways to prevent, diagnose, and treat pandemic influenza. These efforts include testing new drug and vaccine concepts, studying flu viruses in animals and humans, and documenting factors that could lead to the emergence of strains with pandemic potential. The aims of these projects are to help health officials quickly detect an emerging influenza A virus and implement measures that bring the outbreak under control.
Okay, so so you got an idea about what is antigenic shift and drift and how the reassortment happens, right? So there is some overlap on those 2 videos, but I try to play it 2 times, just it's engraved in your brain, just what is happening in the flu and how important it is, because we always overlook this, and especially in India, like huge billions, 1.2 billion. Uh, so some people dying.
1.4 degrees.
Huh?
1.4.
1.4 now? Okay. So 1.4 billion, right? So if some people die, we don't care, but I think we should care, right? Even though one people died, I think that's the humanity. I think that's more important. I think that's why um, it's very important to understand those. Even like government consensus does not record fully. How many are dying due to flu and all those. So a lot of data is missing. Even though in this computer age, so many sophisticated AI technologies coming in, but if still we are missing those informations, right? At least epidemiological side view, that how many are actually infected, how many are died, how many actually took that drugs. Is there any clear report? I don't think so in this country, right? I think that's is important very much to kindly figure it out how many deaths happened and what the government is taking initiative and those things. I think.
So what follow-up will happen, because otherwise it will be a lot more from the present government.
No, yeah, I think. Yeah, I mean, because the flu is very important. I mean, even especially elderly people, right? You saw like the person has a diabetes, some heart disease, and then got a flu. So definitely it will aggravate the situations, and the person may die. This was got a pneumonia and died. Uh, have you looked over more in depth that what kind of pneumonia he got? Okay, he's old, he has to go. I mean, that way people always tell. I disagree at that point. I mean, even though, if he still live for another day, I think it's a human life. So we should be we should have that humanity to, even though they are old, they should have the freedom to live, right? It's nobody's, they're not living somebody's mercy. That my point is. That's why,
One thing,
yeah.
they are serious with respect to some of the diseases, like if you consider polio, TB.
Oh yeah.
They are very serious. Monitor well and.
Yeah, yeah.
And then that's it. But so said flu case.
No, that's the reason. Like flu is also a lot of death happenings, because it's not like Kaisenur forehead disease. It's a very few, right? Chandipura, one or two. It's okay. We don't need that much to emphasis because it's not major. Yeah, but if it because we may not have too much impact, if the COVID does not kill too many people, right? Then people say, oh, COVID is killing people, that's bad. Then people, but flu also killing a lot, but we don't actually track those. Even tuberculosis and malaria and all kind of major things, right? So. Can you please the next slide? Okay, so it is another example to show how the reassortment or antigenic drift happening, shift and drift. So same thing, what you saw in the video is another like maybe in the slide form, like the dog has a lot of viruses, it is going to the pig. Same, the human has a virus, it's going to the pig, and then it has a mutation, reassortment happening, and it is become more pathogenic and infecting this. So here is another example, like a canine flu and human flu. So canine also has a flu. So it has canine flu, and I have a pet, and I have the flu, and I have transmitted to the canine, and then it has become the reassortment, a different kind of virus generated, and that is killing, maybe pathogenic for both canine as well as human too. So there are multiple phases of influenza pandemic. So phase 1 and 2, which is predominantly animal infections, few human infection, that is called phase 1 and 2, 3. And then when it is 4, it is mostly sustained in human to human transmission. And this is kind of pandemic, 5 and 6. So, and then it's a post-peak and post-pandemic. So we can go a little depth, what is phase 1, 2, and 3. Phase 1 is no virus infection has been detected in humans, but because it is mostly present in the animals, so there is no cases detected in humans. When it is no, it's still in a circulating stage in the animals, but there is still no new influenza virus subtypes has been detected. Still, then it calls phase 2. Okay, but there is a circulating animal influenza virus. So that is the demarcation between phase 1 and phase 2. The phase 3, at that point, like human infection with new subtypes, we can see that, okay, I find a humans that has a new subtypes, but there is no human to human spread is there. So that is called phase 3. When phase 4 comes, I can see there is a human to human transmission happening, and then it called phase 4. And we can think that spread is high localized. It's not like everywhere, like maybe in the small part of Kolkata or somewhere it's happening. So that's called phase 4. And but phase 5, as its name's is pandemic, means it's everywhere. It's a broader area it's happening. And phase 6 is also increased and sustained transmission in general population when it is happening. So as Dr. Radio is asking, like, there are different combinations, right? H has 1 to 17 and N has 1 to 10. So definitely there is so many, like 170 combinations right there. Um, so you can see majority H1N1, that is the latest 2009, which is by swine flu pandemic, called H5N1, H3N2. That's mostly. Here it is not too well defined. H3N8, H2N2 is another one. It was a pandemic, but mostly majority H3N2, H1N1, and H5N1. So those are the major. That was your question too. Like, they are the major causes more pathogenic. And based on the death, I think that was the 1918, where more than 50 million people died due to the Spanish flu. That is the major death happened. And then 1957 is an Asian flu. And then Hong Kong. And then 2009, there is another outbreak happened, the swine flu. Yesterday was, Dr. Reddy was asking, it is long ago, there's technology was not that advanced yet, and how people find out whether the RNA, that's the plot I draw, I have shown that, as it more, since 1950 or so, I figured till 2020, there's more RNA species is developing and all those. So the question is, maybe that time there's not enough technology and we have not detected. That's why it is low. But now we have more technology. That's why you can detect. But that is not 100% true. Like, they have shown here the Spanish flu. They have find out some tissues got from the army recruits in 1918 in Kansas, and they can see those tissues. It's an 80-year-old flu, a bit of a long tissue they have preserved. And from that tissue, you can definitely sequence it and find out what kind of RNA sequence. So you can do the whole, genome sequencing, from these tissues, and can get all the informations. And so the bottom line is there are more than 50 million deaths globally happening, and more than 2 million deaths in USA happened. So still ended World War II, World War One. 1918 is the end of the World War One, so still the flu is still present right now. So the question is, why 1918 the virus was so deadly, which killed 50 million? They say that mostly there is those non-structural proteins. They are more avian than human. So maybe they say that it's because it's an avian flu. They are more pathogenic some way, and that's why they graphed 1911 to 17 how the flu was there, and in 18 it's mostly affected very like a young adults 25 to 44. There's a high death rates in those populations compared to the other folks. So probably that came from the the avian flu, which is a non-structural proteins that may be responsible for this death. There are some pictures here, like Hong Kong flu, which killed about 70,000, and President Ford hoped to avoid a repeat from 1976 with the swine flu. So people are taking vaccines here in a queue. Swine flu happened in 2009. The first case reported in mid-April in Mexico and United States, and that is the strain H1N1. The triple reassortment containing swine, avian, human influenza components. So these are the triple reassortment containing. So it came from swine, avian, and human. There's a global pandemic declared on June 11, and official reporting of individual cases discontinued on 2009. So that is another example how the future happened. Secondary attacks rates, US 8 to 8%. It is the predominant strains. There are 21 resistant to the Tamiflu, that drugs. And there is a mutation also they find out. So basically, they, when the flu happened, they try to clean everything, have a mask there. This is a picture of, school. They try to clean every day after the school hours, make sure the benches and chairs and the floor are free from any virus. They clean with the, Lysol or other antiviral agents. Wearing the gloves. So those are kind of precautionary measures also. They should not get contaminated or they got infections. So this is the map how the death from 2009 influenza pandemic happened. And the circle shows how bigger the pandemic on that area, on this country. So it is globally happened in the different. So after that, so all the countries has some of the headquarters or surveillance centers. So they have like a reference center or research programs happen. And definitely in India there are like 1, 4, 5 places there. Their job is to find out what kind of virus it is, what strain it is, or whether it is H3N2 or H1N1. So it is globally distributed, those centers, and they monitor. And that's why I saw you a couple slides ago, like based on that they can determine what vaccines should be given, whether in the northern hemisphere or southern hemisphere or tropical area. So those centers collect those data throughout the world. The regulatory center WHO is right here in Washington, and also the headquarters is also right there in the United States. But there are centers everywhere. They collect different samples. All those patients who got infected, or they do next generation sequencing, find out the sequence, and then send to the headquarters. And then they decide, okay, the next year the virus, what vaccine we should make, and that is the global headquarters. They decide what kind of vaccine they do. You can play that video too. So this video is talking about another country, how they control those, what are the strategies they have taken to control the influenza.
As one of among the priority diseases within the country. We are thinking that the influenza is just a surrogate of many of the zoonotic diseases, and countries, especially in Africa, they need to prepare in terms of really the capacity in terms of detection and the lab capacity too. And we also have the National Influenza Center lab, the NIC lab, which not only does the influenza detection, but it has been used to build also capacity for other epidemic diseases. In 2009, we had an epidemic of cholera. We had also episodes of measles, and we have been using the same capacity which have been built for influenza to address detection of these other epidemics. And that is why we are actually seeing that the capacity which has been built for the NIC, it's actually the capacity which has been built for the overall health system within the country. Before
Before
2007,
2007,
we
we
didn't
didn't
really
really
have a
have a
capacity to
capacity to
detect
detect
influenza
influenza
as
as
well as
well
the
as the
measles
measles
and
and
cholera.
cholera.
So
So
the
the
lab
lab
capacity
capacity
was
was
very
very
minimal,
minimal,
and
and
also the
also the
surveillance
surveillance
capacity
capacity
with
with
the
the
inclusion
inclusion
of
of
some of
some
the
of the
funding
funding
within
within
the
the
PIP. We
we
have
have
actually
actually
used
used
that
that
funding
funding
to
to
now
now
build
build
those
those
pillars.
pillars.
We
We
had
had
cases
cases
in
in
2009
2009
for
for
cholera. We
cholera. We
had
had
also
also
cases
cases
for
for
Rift
Rift
Valley
Valley
fever.
fever.
And
And
these
these
people
people
who
who
have been
have
built
built
capacity
capacity
for
for
influenza and
influenza and
other
other
related
related
diseases,
diseases,
they've
they
actually
actually
used the
use the
same
same
techniques, the
techniques, the
same
same
technology, the
technology, the
same
same
NIC
NIC
capacity to
capacity to
really
really
like be
be
able to
able to
identify
identify
these
these
cases.
cases.
And
And
we
we
could
could
using the
using the
NIC
NIC
lab
lab
as a
as a
whole, we
whole, we
could
could
really be
really be
able to
able to
respond
respond
on
on
time
time
and
and
really
really
reduce the
reduce the
morbidity and
morbidity and
mortality.
mortality.
So basically, it's talking about like how the NIC, which has been developed, which, the previous slide we have shown, this slide, like after that, the pandemic. So every country has their center NICs, and they report to the Washington so that they can come up with the different vaccines. Next slide, please. Oh. Okay, so all those NICs get those data. Okay, what is happening? What kind of strains are available? What happened the last year, and they give those report to the headquarter or WHO in Washington, and then they come up with that cocktail of vaccines. So this is for 2025, 26, which is right now. I think I took that vaccine. And there are two kind of vaccine: eggs-based vaccine and the cell culture recombinant protein or nucleic acid vaccine. So you can take any one of them, and it's covering like H1N1, H3N2, and there is another B influenza B virus there too. Same like H1N1, H3N2, and B virus. So basically, they give this trivalent vaccine to protect, and this find out based on the previous all NIC documents in the northern hemisphere. This should be vaccines we should use for the next year. Same like southern hemisphere, also the same idea, doing the same H1N1, B, influenza B, H1N1, H3N2, B influenza. So I cannot find out for the tropical, but northern and southern hemisphere, they have designed, the WHO has given this recommendations, and it may change for every year, and you have to change, you have to check the WHO website, and they have the informations what the next next year vaccine should be. They normally release before the time, like normally for in northern hemisphere it starts from November. So you can see in the website, I think probably they already list in the August or September what are the vaccine they are going to give today. Yeah, so these are the recombinant, all the recombinant protein. Yeah. Yeah. Yes. You see, they are mostly focusing on those H and N. They don't need those other things. It's most of the recombinant. So yeah, I looked over some such. There is a daily NCR. The H3N2 outbreak happened, and you can that's the report I saw. And also in Japan, H3N2. Recently in October, I saw their newspaper that has been considered as an epidemic in Japan. So that's if you see the vaccine, they're covering H3N2. So definitely H3N2, if you take that vaccine, it will protect somehow. But again, it is there in India. There is some death happened. If you read that newspaper, I got from the Times of India, but also in the Japan they declared, and it came in the Nature paper too. So, ICMR reported, yes, just here, I'm showing here, in 5 viral infection, including dengue, influenza, rising fast across India. This is the ICMR report. I find out from November 3rd document. And India is witnessing a surprise in viral infections once by ICMR. So it's the top disease, and that's why I'm asked initially before I start, like, do you have a flu vaccine yet? So, but I'm not a policymaker. I'm just a scientist. But my point of to inform you that you should be aware. You should talk to your facility doctors, like doctors may know those information so that they're vaccinable and they can guide you where to get it, and if you're really interested. I think that's more important, especially if you are traveling somewhere internationally, or there are prompts like if you are going to Delhi or something, there's flu there already. You should take precautions that way. I mean, or wear mask or something. Take those precautions.
Precautions will be there like this.
What?
Precautions will be there like this. The media,
Exactly.
walking will be
No, that's why as a human being, it's your responsibility too to protect yourself, right? And I don't care. My principle is totally different. I read this, okay, and I take my protections. Even though somebody say, oh, don't do this because they have not declared you have to wear mask. Even when COVID, we are all working. I told my family, everybody has to wear mask despite what the government says, because I know it is a respiratory, and everybody, my daughter, everybody is going to school, then school closed, and it was from in-house. But my technician, my postdocs, and everybody, I said you have to wear mask whether any government official says yes or no, because it is your body. They will not dictate, because you have to take care of yourself first. And the same thing here also, despite government says and they forget, but it's, as a human being, it's also your. I'm advocating that way. As a scientist, I also like to it's your health, it's your body. You have to protect yourself. It's not somebody will dictate you, because you are knowledgeable enough to study more, do research more, and go other papers on what they are doing, what others doing, and take the next step. Right? So as already they have declared an ICMR, which is one of the premier institute of medical research, Indian Council of Medical Research. So they are continuously monitoring what are and their job also to monitor those. I mean, when they are recommending, I don't know, there are a lot of political issues and other things, and how it will be applied to everybody, 1.4 billion people. But at least.
until unless it is going to be predicted as epidemic.
Yeah, exactly. Exactly. That's I said. Like until COVID, people are dying. Then people are conscious. Oh, I have to do something. But before you go that far, you should take initial steps. So that's my point.
So, recently one in Kerala, they say some amoeba kind of infection is happening.
Oh yeah, the brain eating. Yeah.
Still people are going.
Exactly. Yeah.
Only few people are worrying with this. That's it. Unless it becomes severe or unpredictable or cannot be managed that time.
Exactly. I think that's another thing. Like where people awareness should be increased. It's you have fun, you go visiting place, but also you should be aware that that's why I showed the first yesterday I told the Maslow's law. It's a pyramid, Maslow's law. The basic is the necessity. We all want food, shelter. That's the basic necessity. Everybody wants that. Then the next thing is like a safety. What is the safety? Health, employment, education. That's the safety. Because if I am educated, I may get a job. If I am healthy, I can do the good work. So that's the second necessity. And I think these two necessities definitely to be fulfilled. And health is one of them. And then once you finish that safety thing, then you can think about, oh, I need to self-esteem, self-actualization. So I need to be topper. So people should remember me. So that's kind of like a Bollywood stars and those big singers on those. So they are on the top stage. They already covered those. But as a general people, we have to cover those first two steps: necessity and safety. And then you can think about, okay, I'm healthy. I have a good job, I have not to think about food. Now I can think about do some more good things. Self-actualization, self-esteem, those comes next. But as a, you're already, first we are not talking about, right? Otherwise we are not here. I think second also we should be focused on that, because the health, as we are talking, the whole course is about also health, highly related to health. And if we are healthy, we can think about, right? If I'm not healthy, I never come here, right? Though I got sick, a couple days. But that's the point, right? So if you are healthy, you can think about, okay, I'll do masters, I'll do PhD, I'll do big scientist, I'll do research, I'll do more collaboration. But if you are not healthy, forget about it, right? You are thinking, oh, how long I live? A few days left. Those things. So, so that's why it's already reported, and I think you should take care of it. And also, another important is 1 in 9% tested found positive for infectious disease in ICMR. So the top 5 is this, right? And so you can do easily the calculations. Exactly. So it's a not made by me. It's an ICMR report. So they are saying, so you definitely take care of those infections that we already talk about. Dengue, we are talking today influenza, and then I think there are other, this hepatitis A, I think we are talking tomorrow, or I don't think we have a time today, but maybe I'm talking today. Yes, norovirus I'm talking, and there is herpes virus, which is a different topic. We cannot cover this time. So that is another one. We talked about herpes simplex 1, herpes simplex 2, varicella zoster, chicken pox. Those are very important, and every time people ignore it, especially children's. They are very susceptible to chicken pox, and so yeah. So covering hepatitis A, because I put it there. So there is anti-H hepatitis A, B, IgG is high. There are 2 types of this. Either you have a high or medium based on the immunoglobulin. Okay. Hepatitis A disease rate. When the immunoglobulin is low, then there is more chance of this. And age of infection is early infancy, late infancy. Oh. Yeah, I think there are 4 slides. Huh?
4 slides.
Yeah.
9 slides.
Oh, 9 cents, so we can take a break? Yeah. I'm ready.